Hello, good morning, good afternoon, good evening, and thank you for joining us in the last seminar of this series for the International Plant Breeding Seminar here from North Carolina State University. We have the pleasure and honor today of having with us Dr. Gustavo Rodriguez, who works at the Instituto de Investigaciones en Ciencias Agrarias de Rosario in Argentina. He's also part of the National Scientific and Technical Research Council. He is a professor of the Universidad Nacional de Rosario, and he works in breeding tomatoes and using all sorts of genetic resources for that end. Gustavo, it's a pleasure to have you with us. I'm going to stop sharing and give you the floor for you to share.
Okay, thank you,
I see
Carlos.
your screen. I see your screen, and it's good.
Okay, so I will start. Thank you for the invitation, first of all, to participate in this International Plant Breeding Seminar. How Carlos say, I was talking about tomatoes and the use of wild tomato species to increase the performance of the cultivated tomato. Okay, but first I will introduce myself because all that I were talking to you is related with my academic formation and the place where I came from. I am an agricultural engineer, then I did a master degree in plant genetics or plant breeding, and then I obtained my PhD in agricultural science in the National University of Rosario at the Faculty of Agricultural School. All this located in the campus that is 20 miles far from the Rosario city, that is the third most important city in Argentina. And all this is far from the capital, Buenos Aires, to 200 miles approximately. This is our campus that is located here. And if you can or decide or planning to visit us, we can show the campus, but we can show to the Lionel Messi house because it's very close to our campus. You know, the Messi is the GOAT or the most famous player in the world. When I finished my graduate student, I moved to Ohio State University. There I was working with Esther van der Nap that worked in fruit genes involved in fruit morphology. And then when I returned to Argentina in 2007, I get my research position at CONICET and my professor position in the university. Okay, why tomatoes? Tomatoes is not the most important crop regard to the production in tons or the harvest area, but it's one of the most frequent in terms of the productivity per square meters, or is one of the most important, the seven most important crops regard to the income that it's generated. Okay, but within the vegetal crop is the second most important consuming vegetable, and the reason is the versatility that the tomato has to prepare or elaborate meals and food, cook and fresh, and is a source of lycopene, flavonoids, vitamins, minerals, and fiber. That's the reason because tomato is considered a functional food. It's a plant model for genetics for climateric fruits, and it's a model too for breeding of cell crops species. It's a diploid species. The number of basic chromosome is 12 and has a small genome around 950 megabase. The cycle of crop is short, that allow to make more than one season a year. The genome sequence, we have a reference genome from 2012, and until now we have more than 2,000 cultivar or variety that has been sequenced, and they allow to build the pan genome that we have to use in the breeding programs. Maybe you know this information and this data about the production of the tomato in the world. The third or the biggest producer in the world are China, USA, and India. In Latin America and South America, the most important producer is Brazil, with almost 4 million of tons per year. And Argentina is the second most important producer in Latin America and South America, with almost 1.2 million of tons per year. And the average yield is around 8 kilograms per square meters. The consumption in our country is 4 times higher than the global consumption, or the average of the global consumption, that is almost 6 kilograms per habitat per year. These are the productive zone of tomatoes in Argentina. The only zone that is destined to the tomato for industry is the Andean region, and the others are destined for fresh market tomatoes. This zone in the northwest produce tomato for autumn and the start of the winter in Argentina, and the other region are more or are producers of tomato in the spring and summer season. The Corrientes province is the place where the tomato is mostly cultivated under greenhouse, and the rest is most open field tomatoes. Around or near to the biggest cities in Argentina, we have the Hort Belt. This is the case of Rosario, that we have an Hort Belt or productive zone for vegetables where we grow tomatoes. And this Hort Belt give vegetable for almost 2 million of people. And this is a place where we are located, our university or our campus. But what mean when we say fruit quality in tomatoes? The fruit quality is composed by those attributes that are related with the visual aspect, like size, shape, and color, and these attributes are the first property that decide the election for the consumers in the market. The second, or when the market repeat the vote in the market, is if this product has good savor and flavor. And the third, very important for us, is the shelf life. The shelf life is the preservation of these attribute, size, shape, and color, and savor and flavor, from the harvest to the consumption. What more longer is this time, the shelf is a good attribute. What are the challenges in Argentina for tomato breeding? Okay, Argentina import most of the seed that we use, almost 98%. You see in this graph, these are the registration for intellectual property of cultivar in Argentina. In the last 10 years, around 300 cultivars were registered, but only 4% were developed or created in breeding programs in Argentina. And all of them are open pollinated varieties. We don't have the possibility or facility to produce hybrids. So the cultivars that are used in our production system are not adapted to the environment or the type of system that you use to produce. Additional, the breeding programs globally and in our country are more focused in yield, performance, and pest resistance, but the consumer globally is question the fruit quality of these products. So within the fruit quality, shelf life, how I mentioned it before, is a critical trait, because for us, for all developing countries, the total production, the over of the total production is 50% of total production is lost during the market change. For this reason, the fruit shelf life is very important attribute to improve in the breeding programs. But what are the source for genes to extend the fruit shelf life in tomato? This is a graph very known in tomato for the ripening process. The fruit grow after fertilization of the ovary, when by cell division and cell expansion, until the fruit achieve the maximum size, that this is the mature green stage. Then the fruit has a peak of ethylene, and this is the reason because the tomato is classified like climateric fruit. And then start to accumulation, or start the change in the biochemical components in the fruit. One of them is the accumulation of carotenoids, and the rat rape stage is when the fruit is totally with the color of the maduration. After that, the fruit is a leaf organ, therefore this start to die. If we want to have fruits with shell life, we need to extend this period between the breaker, that is when the fruit is harvested in the plant, to the fruit ripe or fruit disease. One of the strategies that is common use in the breeding program is the cross-seed with mutants that are in the tomato germplasms. The most known mutant are the NOR, that's a non-ripening mutant, the RIN inhibitor from the ripening, and the alcobaca. All of them are involved in the synthesis or in the pathway of the ethylene hormones. But the problem is all these mutants in the F1 condition had good or had shell life, but the problem is that they have pleiotropic effect on other fruit qualities like color, texture, or juiciness. The other strategy is to try to use transgenesis or new breeding technology, editing, to try to modify this pathway in the ripening process. You know that in tomato was the first commercial genetically modified food with the flower safe technology. They block the synthesis of the polygalacturonides, that is the enzyme that degrade the cell wall in the tomatoes, but that was not good in the from the commercial point of view. Both GMO on new breeding technology in less extent, but both of them has a issue about the perception of the consumer. More in one country than another. For example, in Europe, take that the editing is a GMO, so it's not allowed to produce tomatoes by this new breeding technology in Europe. Other possibility that is the fundamental of our breeding program is try to use the wild genes, the gene present in the wild species. The hypothesis behind is that if the fruit preserves the good quality, the good color and flavor and savor during a more extensive period, the predators has more opportunity to deceive this person. So this is an advantage, evolution advantage, or change for the adaptation to the wild species in the natural environment. You know that the wild species grow in the center of the origin of the tomato, that is South America, in the Andean region. There grow like weeds, 12 wild species. Only one of them is Solanum pimpinellifolium, that is the ancestor of the tomato cultivated tomato. But two centers of domestication are proposed for tomato. One of them is the same that the center of the origin. The other is the region of the Yucatan in Mexico, where in this period of the domestication, the tomato increased the fruit size. Other shapes appear and different colors. And when Columbus arrived to America, and then the Spanish carried the tomato to Europe, where first were used only like ornamental plant, but then in Italy and Spain was incorporated in the cook. And in this moment start a process of breeding that create in the last 50 years, 500 years, this diversity that we have in tomato for color, shapes, and size, and other attributes that we cannot see. But we know that the only 5% of the total variability present in the tomatoes germ plants is in the cultivated tomato. The rest is present in the wild species of tomato. So based on this, our first step for trying to apply this approach in tomato for extended future life was a work done by Guillermo Prata, this actually colleagues and member of our group, and Roxana Sorsoli and Picardi Liliana, that they were my advisor during my PhD and my master's degree. And what they do, they make crosses between several Argentinian tomato cultivars, several wild species, and 2 mutants for ripening, nor and green. And what they found that the F1 between the cultivated tomato and the mutant has similar fruit shape average value than the crosses made with wild species and cultivated tomato. But the advantage of this last cross is that the fruit quality was better. I mean, the fruit was small, but the color, texture, juiciness, content of sugar was high than the cross with the mutants. And this opened a possibility to start a breeding program to obtain tomatoes with longer fruit life. Based on this pioneer work, we select 2 parental for our breeding programs. One of them was the Argentina cultivar Caimanta, obtained by the INTA Cerrillos in Salta. Here you can see the pedigree of this cultivar that includes some, in the pedigree, some cultivar originated from Argentina and other from America, like Flor Date or West Virginia. This cultivar was a cultivar for fresh market, has a good health profile. The fruit weight was around 100 grams, and the fruit life very small or very short is around 10 days. On the other hand, the wild species parental genotype that we incorporate in our breeding program was LA 0722 from Solanum pimpernellifolium. This was collected in Peru, in Trujillo, has indeterminate growth habits, very round fruit, very small fruit, around 1 gram, but the shelf life is around 20 days. And making cross between this parental genotype, we follow 2, sorry. First, now we have the sequence, the genome sequence of these 2 parental genotypes. Here you can see the SNP density and the indel density map regard to the reference genome in tomato, the Haynes 1706. And when we compare the genome between both parental genotypes in our breeding program, we found almost around 1 million SNP between them and around 300,000 indels between them. And this information we use for develop molecular markers and make studies about diversity, develop genetic maps, and detection of QTLs in our populations. Now, based on these 2 parental genotypes that we select, we use 2 breeding strategies in our breeding program. One of them was the obtention of recombinant inbred line. We know the recombinant breeding line is the select some F2 plant, and based in this case was based, or the criterion of selection was based in the shelf life traits, and the selected plant was selfing during 7 cycles. At the end of this process, we obtain inbred line and recombine at random the 50% of the genome of each parent, but they are established lines. The other approximation or approach that we follow in our breeding program is the mutation of the near isogenic line. For this, we use 4 backcross after obtaining the F1 hybrids. And the purpose of this was only obtain a cultivar very similar with Caimanta, the cultivar Caimanta, but with individual introgression random in the genome that came from the wild species parental. In this case, in this approach, the selection of criterion was the fruitful life, and the advance in the backcross was assisted by SSR markers. Now I will present the result about the real collection. This is the F2. The F2 between these two parental genotypes was about or around 100 plants, and this is the histogram or the phenotypic distribution for fruit weight. You can see here that the distribution is skewed for the value similar to the wild species parent, and when we see the distribution for the fruitful life, we see that some F2 individual has more or higher value for shell life than the wild species parent that was the parent with the higher value for this trait. That means that we found transgressive segregation for this trait in the F2. Here I present the B plot between the relation that graphical is the relationship between both traits in the F2 plants. In a traditional or commercial plant breeding or tomato breeding program, we only select the plant that have high fruit weight and high shell life trait that is in the group 1. But our breeding program is an academic breeding program. We are interested in trying to understand what is the inheritance of these traits in the tomato germ plants. So we select in an antagonist and divergent way plant from the 4 groups in this B plot. This was the criteria, the 4 combination, and after 7 cycles of selfing, we obtained the 18 reels, and this is the reels ordered by fruit weight. One of them was or present or show yellow fruits, and this is the blob for this trait evaluated during 3 seasons. The first one was evaluated to field open experiment, and the other 2, these plants were evaluated under greenhouse. And you can see that the most of the plant, or all the plant, or non-plant, or non-real, actually, achieve the fruit weight of the cultivar Caimanta. That means that all these type of reels are fruit like more like cherry tomatoes for the fruit size. The heavier reel was the reel 14 with the fruit weight around 30 grams. And the broad sense interactivity for this trait in this experiment was around the 70%. On the other hand, in this graph on the right, you can see that the fruitful life has an inherent broad sense heritability around 50%, and you can see now the blob, the reel with more or high number of shell life is the reel is 17, the yellow one, but we have several reels that achieve values higher than the wild type tomato. Here's like a Manhattan plot. We run some molecular markers, indels, and SSR through all the genome in tomato, and this is the association. About this line is the significant association. Most of the association were found with fruit weight that is here is shown in black dots. We find association in some non-QTL for fruit weight, other was a new association on association that were not reported before. But for Fruition Lab, that is shown here in white dots, we find one association with Fruition Life in the chromosome 10 with the probability less than 0.001 or 1%. And these two association was at 5% significant and was one in the chromosome 9, the other in the chromosome 11. Okay, so we found in this collection variability for fruit weight and fruit life. Some of the lines or recombinant hybrid life has more than fruit life than the wild species parent, and that was good for us. Then we advanced in this type of study where we characterize the collection or the real collection for metabolite content. In this case, the metabolites were detected for or based on two methods. One was GCMS and the other was MNR, magnetic resonance nuclear. In this graph, you can is displayed the number of detected metabolites compared with the cultivated tomato or caimanta in this case. You can see here, this is a metabolic profile for the Solanum pimpinifolium, OLA 07022, and you can see that the metabolic profile is totally different to the cultivated tomato. The one is has intermediate value or an intermediate profile, and in the real collection you have diversity or broad variability, and several of them are more similar in the metabolic profile to the wild species than the cultivated species. This is the color represent different groups of metabolites. The green is amino acid, the blue is organic acid, etc. But within the metabolite that we detect with the NMR, one of them revised some reference or papers. We found that one of them is related with, or we can propose this metabolite like a nutraceutical metabolite because this chemical compound was found associated with the prevention of metabolic syndrome that is related with the type 2 diabetes. And we see this variation in our collection for this metabolite. You see here is the metabolite detected in the wild species. Only trace are found in the cultivated tomato. This metabolite was evaluated in other cultivated tomato and the cultivated tomato that arrived to the store. And this metabolite only found in very small quantity, but some of our lines has 400 more content than the cultivated tomato. And based on this result, we started an explorative experiment where we select one rat line that is present metabolic syndrome and obesity, and we feed it with food supplemental with the tomato with high level of metabolites. In this first experiment, we see that the body weight and the circumference in the abdominal and thoracic part was a significant decrease in the rat treated with the tomato with the metabolites, but we didn't see any clear association with serological parameters. We are repeat this experiment in a more complete design, and we are expecting this result. But This first experiment says that maybe our lives is have additional functional properties than the tomato has itself. This is the another approximation approach to study and to develop future life cultivars, cultivar with longer future life. And this is the approach that we use is the near isogenic line developed. I say that we make 4 bacrosis from the F1, and this is the image represent the fruit of each line that this collection of 22 Niels were evaluated in 2 essays during 2 seasons, and this is a result presented in a principal component analysis that we group these Niels in 4 cluster of groups. One of them is more associated with heavier fruit weight or heavier weight fruits, fruit with more acidity, and this is the group that more important for us, that is associated with fruit with longer shelf life, associated with high content of sugar and some parameters of color. And with the molecular marker, we ran to try to associate the polymorphism in the genome with the phenotypic values. This is the results are present in this heat map, and you can see here that these are the fruit related with the size and shape, and these are the fruit related with the fruit quality, and all the squaring color mean that we found as significant associations, and red color mean that the allele decreased the mean value of the traits, and in blue means that the white allele increased the mean value of the traits. And you can see here that for most of the attributes related with the fruit size, the white allele decreased the mean values, and for the fruit quality attribute, the white allele increased the mean value. This is fruit life. You can see several associations around throughout the genome of the tomato that are associated with the good values of performance for shelf life. From this collection of Niels, we select 2 because they have promising attribute for fruit quality. One of them was the near isogenic line 115, other than 080, and they have high ferment or high fruit shelf life compared with the cultivated tomato. This is showing this radar graph or radar maps. And we analyzed the transcriptomic of this Niels in 2 ripening stages, mature green and red ripe. And then we analyzed also the differential genes in a proteomic analysis that is showing this graph. We found several differential genes expressed gene in both in transcriptomic and proteomic analysis. Based on these 2 results and integrate them, we can propose some gene affecting fruit ripening that are present in our Niels collection. Some of them are related with seal wall remodeling, other with the ethylene pathway, other with the biotic and biotic stress, and this last one with the metabolism. For us, the most interesting result found here is the integration of the synergy between these 2 genes, the apetalate-2, that previously was only reported for develop some organ in fruit, but not for fruit ripening. And this gene is molecular associated with this kinase gene. Okay. Based on these results, we select some cherry type tomatoes and and two late tomatoes to make the registration of proper intellectual property in our government office, and these are available for seed commercialization in our country, and this is in process now to make the registration. But in our country, it's so difficult to try to commercialize open pollinated varieties because the law protects to the gardeners of farm. They can save the seed, and they can save the seed without paying any fees or licensing. But this is the reason because in 2019, we signed an agreement with the municipality of Rosario that this is the city of Rosario, and the municipality groups the gardeners or producers that are distributed inside or in the urban region and the peri-urban region in the city. All them are gardeners that produce tomatoes. In the last year, the law in Argentina changed. In this region, the urban and peri-urban region, the application of chemical products for control of pests and disease is not allowed. So they was needed to transition the production of this production system to more organic or agroecological production. For us, this type of production is a good candidate to try or to test our plant material in Rosario. So we contact some of these gardener or producers in Rosario that they sell the production in local fairs. We give them some seedling of our plant material together with the survey, when they can record the productivity, the health condition or health profile, and the acceptation or not of the plant material in the local fairs. This is for us a good approach to test our plant material in this type of product systems. When we do this type of activity, the acronym that we use is Código Tomate or Tomato Code. That means that the conservation and divulgation of tomato germ plants in our city of Rosario, our group is known like Tomato Code, not like tomato genetic and breeding in the university. Okay, based on this agreement, we carry out 2 meetings with local farmers. The first one was made in 2021. In this meeting, we show all our plant material in our campus, and we collect information about what they or the farmer think about the materials, and if they are interested in test some of them in his gardens or orchards. In 2022, we did other meeting in the office of the municipality of Rosario. In that opportunity, we interchanged experience about the previous season with our plant material, and we give new seedlings with other new contacts of producer in the region. This is a work that I are still working in our community. The last part of my presentation is a new research launch in our group. It started the second semester of the last year. It's related to try to search genes for tolerance to salt stress in tomato. In our region, the water for irrigation is taken from the aquifer Welche. This aquifer has water suitable for irrigation for extensive crops, but in the margin or in the side of the Paraná River, where is located our Hort belt, the type of soil that's argidal, the continuous use of this type of water is restricted because increase the salinity in the soil and the soil ratio that is a problem for the production. So we know that the wild species is a good genetic resource to try to search this type of gene or gene tolerant to soil stress. And we know that the Solanum galapagense and Solanum chesmani, that they are endemic for the Galapagos Island, have been reported to be tolerant to salinity and drought at draw. And for us, was very interesting because in the 19 decades, Fulat made several working with our line, or the same line that we work for Fruition Life of Solanum pimenifolium, LA07022, and he reported that this line is tolerant to salinity too. So the first step that we decide to do is start to confirm this result in our group, and we start to make a germ plants collection with plant material conformed by cultivated tomatoes coming from different parts of the world, and some wild species tomatoes. One of them is the LA7022, and the other line is LA1141, originated from Galapagos in Ecuador. And this line was provided by the group of David Francis in Ohio State because it's reported like tolerant to salinity, and David Francis developed a lot of segregated population, and that would allow us to try to map QTLs or candidate gene for salt tolerance. Okay, we plan to evaluate the salt tolerance in 3 crop phases: germination, initial vegetative growth, and whole crop cycle. We only have result for this step, germination. We finished this step. Now we are working in this second crop phases, and we applied 6 treatment. The T0 is embed or irrigate the plant with distilled water. The trait 1 is irrigate the plant with water from osmosis system. The T2 is irrigate the plant with the water taken from the aquifer. And the T3, 4, and 5 are water with prepared solution with different concentration of salts. These are the first results. You can see that we found a variability for germination among all the genotypes that we evaluate in different salt concentration. And the T3 and T4 treatment or salt concentration allow us to discriminate the genotype according to high and low tolerance. In this group, you can see that the decrease of the line is lowly compared with the genotypes classified like low tolerance to salt concentration. And within this group, we found the LA7022, that is our parental genotype. So this is good for us because maybe we can map or try to localize candidate gene in our breeding populations. Now we are move this experiment to the other crop phase, that the vegetative growth. This is our breeding camera where we grow our plants. After the grow, we clean the wash the roots. We take the fruit, the fresh weight and the dry weight of the plant. Then we take image to evaluate roots and aerial parts to determine area and color in the aerial part to demonstrate or to find if the salt concentration affect these parameters, but this is all in the progress. Okay, in conclusion, we demonstrate that, we know that, already know that the exotic germ plants or the wild species tomatoes are useful to get some trait that is not present in the cultivated tomato. That means that the wild species background or germ plant is variability that is still underexploited in the tomato. For us, LA 7022 from Solanum pimpernellifolium provides or give or has or carry out genes to prolong or extend the shelf life in tomato without affecting other fruit quality. And finally, this is our research in progress. This line of Solanum pimpernellifolium could provide gene to develop tomato cultivars with additional functional attributes and tolerant to salinity. Okay, to finalize, this is our group. This is Guillermo Prata, Valentina and Paolo. The three people here are working in bioinformatics. They are trying to study the small heat protein in the tomato germ plants and how they affect the diversity of fruits in tomato. Then we have to Javier Pereira Acosta and Franco. They are working in the project of the near isogenic line. Then now I trying to make crosses between them and try to study in single or in combination the integration, how the integration affect the fruit quality. Celeste, Dana, and Federico are working on fruit shape morphology or fruit morphology. Vladimir and Emiliana are working in the effect of the reciprocal cross in tomato fruit traits. Finally, Nicolas is working in the functional tomato functional food. Tomato has a functional food, and Vladimir and Maria Emilia Baez was included or was hired in our group, are working in the last part, that is the searching for salt tolerance in tomato. So this is our funding, all local or national funding, and thanks for the attention, and I open for questions.
Thank you, Gustavo. Nice presentation. Please post your questions in the chat. While we wait for other questions, I listening to your results in terms of shelf life. First of all, are you hopeful that you can separate those genes from the size, the genes that determine the tomato size? Because it seems that most of your lines that have good shelf life are relatively small in size. And the other question related is, how about flavor?
Okay. Yeah, the first part of the question, yes. The first strategy, we find line with longer shelf life, but the problem is that we can recover the fruit weight in the cultivated tomato. That is the reason because we initiate the second approximation, and now we have some big tomatoes with fruit shelf life. So they are two independent traits. We can select both training. They are not genetic correlated. Okay.
Okay.
And the other plan. Okay. We only we did some panel about our lines in Rosario. We compare in the panel the two lines of our program, GEMA, FSA, and Kerubin, the two lines that are registered. And we compare this line with other that the one cherry line that is in the market, another more Halum tomatoes. Okay, you know that the flavor is a subjective opinion. Some consumer like tomato, more acid tomato, others prefer more sweet tomatoes. Okay. In the preference of the consumer, we see that the best was the line that was present in the market, around 28%, but the two lines that we evaluate, take it together, have more preference than the present in the market. And these two is more better, or is better, they were better than the Halum tomato cherry. I don't know if you.
That's good. Yeah. Good. Well, that's a positive. Yeah. There's a question in the chat. I'm wondering about how you select the germplasm tolerant for salt stress. She said you show a table showing different tomato accessions. Is it based on the origin? How you select originally the materials that you're going to test?
Okay, the cultivated tomato we select from different parts of the world. One of them from American breeding programs, other are heirlooms, another came from the German plants bank from Valencia, because they, okay, we select according to where is the origin of this accession in the German plant bank, because we can infer that if they grow close to the sea, they can take some genes tolerant to salt. But we don't have any report about salt tolerance in the cultivated tomatoes in our collection. On the other hand, we select for the same criteria in the wild species. For that, we sum the 3 wild species that were reported like salt tolerant, and one of them because was collected in this desert area, and we suppose that they can carry gene involved in drought tolerance. Okay, we confirm the LA7022 in the germinate stage, but we confirm the same in the initiate growth stage that are tolerant to salt tolerant. Other that is good for us is Galena, that has a good response to salt tolerance in the.
So yeah, it be interesting to see how the performance is in the following stages, because I imagine that if you can correlate tolerance in later stages with germination tolerance, that will facilitate your screening. On the other hand, if you cannot, having tolerance to salinity in germination may not be of high value, because the most important part of the crop is the vegetative stage. A couple more questions we have.
That was a question that we said. Why Lavando, when he started the study as he did, the report only that we found is until 2000. So what they don't deal with this type of approach. Okay, talking with Dave, they say, okay, in America they don't grant because we don't have you don't have problem with the water. In the because all irrigation is water came from.
So
try to validate the result we found in the initial part with the part the concrete or the wall growth of the
So related to this talk, there's a question about the thresholds for salt tolerance. When do you say something is tolerant? Does it have to have 100% germination? Does it have to have 100% survival?
Yeah, that's a good question. We don't know yet. We define this, but okay. We see, for example, in the late 2022 that the decrease in the T3 is high level or size compared with we find in the water for irrigation, and only decrease in the T3, I think 20%. So
Okay.
if this behavior is constant, you can see, okay, you can increase the number of seeds that you sow in the seedlings.
Okay. Dilip is asking about diseases that may be important for your program and for the farmers in your region.
Yes, okay. We don't evaluate in direct way the disease resistance. It's indirect. We remove plant that have some kind of disease in the field station, but we know that when we evaluate our lines in the Rosario community, they say that the GMA FSA has a good health profile compared with other cultivars that I tried. But yes, we don't evaluate in the direct form the resistant genes.
Okay. Yeah, there's another related question to that about the health of your materials. There's a question back there that says, how are you performing the flavor assessment testing? Is that through panels also, the flavor?
Yeah, we did a panel around 100 consumer. Is the panel you use is not training panel. And we prepare some survey with some specialists that we have in our institution for this type of study. And they have a lot of type of question in the survey. The result that I told you is the last question that I remember, that is the what of 4 tomato you prefer. But we have a lot of data. We know that the preference is not related with the content of sugar, it's not related with the content of acidity. It's more complex than that.
Yeah. Okay. All right. Well, thank you, Gustavo. Thank you for closing this series of seminar and for accepting the invitation. I was wondering, with just a final thought, I do communicate a lot with the Institute for Biosaline Research in Dubai. I don't know if you have any collaborations with them or any exchange, because I can put you in contact with them. I know the director of research, and they're very interested in things like this, okay? Salinity is one of the growing problems in agriculture, okay, because of the repeated use of irrigation, water, and evaporation, okay? So thank you so much, and I will do that. Thank you for your presentation. Thank you for the audience for your participation. Look forward to seeing you all in September when we open up the 8th International Plant Breeding Seminar Series. In the meantime, I hope you guys have a good summer if you are in the Northern Hemisphere, a good winter if you're in the Southern Hemisphere, and enjoy it, and see you soon. Thank you so much.